Good morning, good afternoon, good evening, depending on your location. I am Kaushal Trivedi, Business Development Manager for Water Platforms at Teledyne ISCO. Thank you for joining. The topic of today's webinar is tracking COVID-19 in wastewater. In last 6 months, COVID-19 pandemic has changed the world. More than 8 million people tested positive, and close to 500,000 people died, and thousands of active patients are fighting to get recovered. And these numbers are still growing. Doctors, nurses, ambulance drivers, and all health providers are trying their best to provide each patient a timely and proper treatment so that they can recover fast. To stop the spread, central and local governments are implementing different programs such as social distancing and quarantine, etc. Some countries had implemented country-wide lockdown to control the spread. However, these programs have negative impact on the economy. So many businesses are closed, travels are halted, unemployment is skyrocketing and whatnot. It is challenging to find a good balance between negative impact on economy and keeping virus spread under control. In this gloomy environment, wastewater analysis has provided some hope. In last, I'm sure you all must have come across several news articles about wastewater analysis initiatives taken by cities, universities, and others. It is because the recent findings suggest that virus passes through human bodies and end up in wastewater stream. Also, it remained there alive for a long period, longer than on the hard surface. The RNA analysis of wastewater can detect this presence of virus. Our next presenter will provide more information on this analysis, so I will not go into detail here. However, I would like to mention few benefits of such analysis. If such analysis is done in sewer, close to neighborhood, industrial area, or commercial area, it can help detect presence of carriers in community, even if they are asymptomatic, which can help manage resources proactively. And when I say resources, I mean test kits, test facilities, hospital beds, ventilators, etc. It can also help in making decision whether to implement any program such as social distancing. If such wastewater analysis is performed at hostels and dorms, schools can be made a safer place for students. It can also help boost parents' confidence to send their kids to school. If such wastewater analysis is done at the treatment plant, it can help verifying that treatment process is adequate to kill the virus. It can also help ensuring that treatment plant's discharge is 100% virus free. I can go on and on for such benefit. In short, any type of early information or indications will help us better manage this pandemic and hopefully reduce potential threats in the second wave. Without taking much time, let me introduce two presenters of today's webinar. Our first presenter will be Newsha Ghaeli from BioBot Analytics. BioBot Analytics is located in Cambridge, Massachusetts. They are leader in wastewater epidemiology. It is the first company to commercialize the data from sewage. Newsha is the president and co-founder of BioBot Analytics. In her presentation, she will give an overview of their work on wastewater analysis and how to implement results in a public health initiative, which is very, very important. Our second presenter will be Steve McManus from Teledyne ISCO. Steve is a senior product support specialist with close to 4 decades of experience on Teledyne ISCO water products. For those who don't know, Teledyne ISCO is the first company to introduce wastewater samplers in 70s and is a market leader since then. We offer variety of permanent, portable, transportable samplers for wide range of application. Steve will present samplers and options for the COVID-19 sampling depending on the location. Lastly, please note that BioBot Analytics and Teledyne ISCO are independently operated companies. BioBot Analytics can work on samples collected by any make samplers, and samples collected by Teledyne ISCO samplers can be analyzed by any lab. This is a purely educational webinar where both companies will share their experience to help you make proper decision. With that, I invite our first presenter, Newsha.
Thank you for that introduction. My name is Newsha Ghaeli, and as Kaushal mentioned, I am the president and co-founder of BioBot Analytics. BioBot Analytics is a wastewater epidemiology company, the only company of its kind, and we are building a global human health data asset to really help us stay on top of global public health priorities. And we're doing this by analyzing sewage. At BioBot, we see sewage as an important source of public health information. So if we look at sewage as being a collective urine and stool sample, then we can start to look at human health indicators to provide critical information on the health of our communities. By analyzing sewage, we can monitor infectious disease outbreaks, such as the novel coronavirus, influenza, or Zika. We can understand antibiotic resistance bacteria that are present in our communities. We can look at asymptomatic diseases, such as hepatitis C, map nutrition and diet disparities in our communities. We can also look at pharmaceutical drugs and illicit drugs that are commonly consumed, as well as other substances relevant to public health leaders, such as nicotine and cannabis. BioBot was founded in 2017, but the founding team has been doing research in wastewater epidemiology since 2013. My co-founder, Dr. Mariana Matus, completed her PhD in computational biology at MIT, where she specialized in the field of wastewater epidemiology. Her dissertation has really set the foundation of the company's technology development. My background is in the implementation of smart city technologies, and our scientific co-founder and director, Professor Eric Alm, is an MIT professor of biological engineering, leading the Center for Microbiome Informatics and Therapeutics. So let's quickly dig into how this works. First, we can either collect wastewater samples from a treatment facility, such as this one, or throughout the sewage network within cities, so that we can better map the specific public health needs and properties of a particular neighborhood. So let's take a look at this video quickly, and if it is a little choppy, that's your internet connection and not ours. So when collecting samples from within the sewer network, we first start by mapping out the network, and we overlay that wastewater network with land use data and census data, so that we know not only where to collect the sample, but we also know the upstream catchment area and the corresponding demographic characteristics of that catchment area. This information is valuable to public health officials when designing specific community interventions. It's important that the samples are collected using a 24-hour automated sampling device, the details of which we'll get into later in this webinar, because then we are confident that the sample accurately represents everybody living in that catchment. Our assumption is that everyone will use the toilet at least once, if not more frequently, over the course of a 24-hour time period. The samples are then transported back to our labs, where we have the ability to look at viruses, bacteria, and chemicals that are harbored in our bodies. In 2018, we launched our first product, addressing what was really the biggest public health crisis in the United States up until about 5-6 months ago, the opioid epidemic. So I'll just very quickly give an overview of the work that we did with the opioid product. But with this product, we were generating data, neighborhood level data on drug use and treatment updated every month. So we measure about 30 different drugs, such as prescription opioids like codeine and tramadol, illicit opioids like heroin, fentanyl, and substitution treatments like methadone and Suboxone. And you can see here in an anonymized map from one of our municipal customers, we collected the samples throughout the sewage network in order to break down priority drugs from neighborhood to neighborhood to help local governments know where to focus limited resources and attention. Our first customer, the town of Cary in North Carolina, successfully decreased overdoses by 40% in the first year that we worked together. And they really credit this success to having had the right data, enabling more appropriate interventions and public health programming. So now let's switch gears for a minute. In February of 2020, BioBot expanded its work to consider the rapid deployment of a COVID-19 product. It was around that time that it was found that the SARS-CoV-2 virus, the virus causing COVID-19, was excreted in the stool of infected patients. And so we knew that the virus RNA should also be found in sewage. We kicked off a research program to develop the methods to detect the virus and estimate COVID-19 prevalence in a community. We launched a research project with collaborators from MIT, Harvard Humanitarian Initiative, as well as the Brigham and Women's Hospital in Boston, and we're the first team in the US to detect the virus in wastewater. In fact, our initial sampling in Massachusetts found that the SARS-CoV-2 virus concentration in wastewater was much higher than what clinically confirmed cases suggested on the sampling days. This really suggested to us that there were orders of magnitude more cases than what was clinically confirmed. And the results of that study can be found in the paper here in the screenshot on the screen. Understanding that this data could be transformative in guiding how we respond to the outbreak, we decided to launch a pro bono campaign with the goal of soliciting samples from up to 100 treatment facilities across the country. We were completely overwhelmed with interest, and ultimately we ended up working with about 400 locations in 42 states. These sites represent over 10% of the US population, and all of them were sending us samples on a weekly basis. So with only 400 samples, we were generating weekly COVID-19 case estimates for over 10% of the US population. The interest was so high for this that we've since decided to launch Wastewater Epidemiology as a service starting in June 1st. This data is particularly valuable for understanding how the virus is trending in a community and determining when to safely reopen and keep open our communities and businesses through frequent and longitudinal sampling and trend analysis. It will act as an early warning to detect the reemergence of COVID-19 in a population so that we can rapidly take action and contain new outbreaks. And it also allows us to test at scale. So for a fraction of the cost of mass individual testing, we can get an overview of the scope of the outbreak in a community. This is our current workflow. So 24-hour composite samples are collected either at a wastewater treatment facility or in-stream from the sewage system. And the samples are then transferred from the collection device into our sampling kit. These kits contain 3 50-milliliter tubes, temperature control, and prepaid shipping labels for next day delivery. The samples arrive at our lab the next morning and are then processed in the lab, and then a report is generated and returned to the communities that we're working with. This entire process takes between 3 to 7 days, with our target turnaround time being on the lower end of that. Just a note here that we are detecting the genetic signature, so the RNA of the SARS-CoV-2 virus, using qPCR. We're using the same primers developed by the CDC. And I also want to highlight that this test does not give us information on viral infectivity, because we are detecting the genetic fragments of the virus. Our results are then communicated as a viral concentration, so copies per liter of sewage. Our report looks something similar to this, and in addition to that viral concentration, we're providing a COVID-19 case estimate based on our latest case estimate models. So the number of individuals that we estimate that have infected stool in a catchment area. We also contextualize the data point to your previous samples, so the previous samples collected from that location, providing that longitudinal trend analysis, and also contextualize the data to the thousands of other samples that we've analyzed for COVID-19 to date, to provide a geographic contextualization. BioBot's work is really very comprehensive when it comes to wastewater epidemiology. So we start by working with communities on the program design. So understanding where and when, what frequencies are most appropriate to sample. So designing and developing appropriate sampling programs for your needs. We support with the sample collection, providing the sampling protocols and mail-in kits, and also connecting you with the folks that we know, such as Teledyne ISCO for hardware. We then do the lab analysis all in-house, as well as data analysis. So leveraging our data science models that have been built with the thousands of samples we've worked with to date, and data interpretation. So through data visualizations and weekly webinars, we do provide expert customer support in understanding what the data is saying. And also, we make available our other product lines to all of the communities that we work with. So whether it's the opioid product or additional products that we have in R&D, such as other types of infectious disease, influenza, Zika, but also antibiotic resistance bacteria. And I will end with a few slides really highlighting that we have all of the necessary expertise, protocols, pipelines to begin testing with any community right away. So we have scaled up our labs to focus and our team to focus 100% on wastewater epidemiology. We have sample collection protocols that have been implemented across hundreds of locations in the country, transportation protocols and relationships with shipping providers accustomed to transporting hundreds of wastewater samples to us, our custom manufactured shipping kits and all the materials required for the kits, established laboratory assays to extract the RNA of the virus and run the qPCR assays that have been tested and optimized over the thousands of samples we've worked with to date. We have established QA/QC controls, a computational biology pipeline to identify the SARS-CoV-2 strains, and always access to our latest data modeling pipelines to convert quantitative estimates of virus concentration to COVID-19 case estimates. And then results that are communicated via a report with data contextualized to all of the samples that we've worked with thus far. The last slide, which I'll leave on the screen before handing it off to Steve, is our values as a company and our mission. Our mission is to transform wastewater infrastructure into a global public health observatory. Today we're working to generate data on the novel coronavirus, and tomorrow it'll be something else. And as our public health priorities shift, so will BioBot Analytics. We believe in data for good, that sewage is really offering us a unique opportunity to make public health more data-driven and effective. We believe in collaboration, that building cutting-edge technology to positively impact our work requires collaboration across many disciplines, as well as many different departments and agencies in our communities. And we also believe in privacy and transparency, both internally and also with our customers. And so with that, it's my pleasure to hand it off to Steve McManus. Thank you.
As Kaushal said, good morning, good afternoon, good evening. I would like to welcome everybody who's taken time out of their busy days to attend and view this presentation with all of us. We thank you for your attendance. This is one of those cases where sometimes it's not always clear of the value or the benefit that is gained from doing sampling. This is one of those cases where one is able to see the value of the purpose be put and applied for the sampling and thus gain the benefit right away. There are a number of reasons why sampling may be performed in the field. It could range from collecting water samples for analysis on sedimentation or solids in the flow, such as construction runoff, stormwater monitoring to define what is being washed off the streets, the parking lots, the buildings, and being introduced into stormwater flows that eventually will make its way to rivers, streams, lakes, ponds, whatever. Monitoring for pathogens, monitoring for metals and other constituents of the like that may be in the flow, chemical constituents, BOD or COD analysis, and a variety of other purposes. The message here is that sampling is used for a variety of purposes. These could be due to regulatory reasons, for research, for monitoring processes, or in cases like this, to be able to apply the knowledge that is gained from the monitoring to improve the health of the area that is being monitored. Each sampling purpose has its unique approach criteria to meet the needs. This could mean cases where we're doing single bottle or composite sampling or multiple bottle sampling, and the value of the sequential versus the composite sampling gives you a lump sum value of the entire day, typically, of what was in that particular flow source over the entire day. Whereas for sequential sampling, the samples are segregated in specific time increments where each bottle may represent an hour's worth of time, so that the individual performing the sampling can determine the trend over that day of whatever constituents they are looking for. We could have sampling that is paced based upon time, where we collect even increments of flow or of sample every 15 minutes, every 30 minutes, whatever the designated interval is over that day. Or we can do flow-weighted sampling, where the intensity of samples collected increases as flow goes up and decreases as flow goes down. There may be consideration for special configuration options, such as pause and resumes, where we desire to collect over specific time periods of each day or specific days of the week. There may be parameter monitoring based upon pH or conductivity or other measurements. Those measurements can also be used for triggering the sampler into operation. There may be data logging, where we want to log that data for monitoring what is going on in the source and tie that to the analysis of the samples that are collected into the bottle. There may be concerns about transport line materials, and this is anything that the water is in contact with from the source to the bottle. We may want to look at vinyl versus Teflon suction line. Are there metal components in the line okay to be in contact with the sampler or sample? For example, if we were doing metal sampling, we would not want to have any metals in the transport line to contact that sample. The container of the bottle itself, do we want glass, polypropylene, or polyethylene material bottles? The message here is that the purpose for sampling will impact the selections of the sampler based upon what its capabilities are, the sample transport materials, the program configuration capability, and other items. That is the place to start before purchasing a sampler, is to ensure that the unit you're looking at for purchasing does what you need it to do, as well as down the road. So for example, today we may want to do composite single bottle sampling, but maybe we have the need down the road where we might want to consider a sequential configuration. In those particular cases, we would probably want to look at a sampler that has the ability to do both. Sampling for COVID-19 is no different than other sampling needs, in that there are some things that we need to consider. COVID-19 is a pathogen, and there are unique requirements for this sampling need. First of all, the whole process begins with the sampler that's in the field, and the way we achieve this to collect those samples is with automated samplers. Automated samplers can ensure the consistency in the collected sample, whereas manual samples are highly subject to variability depending upon who and how that sample was collected. Consistency is critical. Different auto samplers have different capabilities. Again, knowing what you need to do now as well as what potential needs down the road will aid you in ensuring that the selected model is right for you. Do we want composite or sequential, or both? And as Newsha indicated, our typical approach for COVID sampling is composite. Do we want to do flow-paced sampling, or event-based sampling, or time-based sampling? Typically, we'll be doing time-based sampling for COVID. We need to consider the suction line and the material for our bottles. Do we have need to refrigerate those samples? For the sample frequency, will we be pacing the sampler based upon time? In this particular case, again, most samples for COVID will be time, and a good normal approach is to take a sample every 15 minutes, which is 96 samples at the end of the day. Will we be pacing the sampler based upon flow? If we will, or if we do require flow-based sampling, it requires us to have a means to measure flow on site. So is there a flow meter at the application where we desire to collect samples? Something needs to measure that flow to be able to provide that information to the sampler. If it's an external flow meter, we need to know what type of signal that meter provides and what that signal represents. Common signals for from flow meters are 4 to 20 signal, where 4 milliamps represents 0% or zero flow, and 20 milliamps represents 100% flow. And that could be based and varied from application to application. The 6712 sampler can have 700 series flow modules attached that provide this capability. common goal for flow-based samples is to design a program that if we have an average daily flow volume day, we will collect 96 samples. And that is again derived from that time-based rule of thumb for 1 every 15 minutes. So if we have that need to do flow-based sampling, there is a requirement that we have a reasonable, consistent daily volume. Obviously, there's going to be days where it's going to be higher and there's days going to be lower. But as long as that end result is within a relatively consistent value, this is very achievable. However, if we have an application that has a great range of daily volumes, where one day we may have 20,000 gallons of flow, and the next day we may have 1.2 million gallons of flow, it becomes extremely difficult to design a sampling program to be able to successfully sample in that application. That's an application where we're probably going to be required to do time-based to achieve our goal. There are a variety of sampler options. They can range from permanently located refrigerator samplers that are AC powered, and again, because of the size, they're just not intended for portability, so they will be in that same location day in and day out. We have a variety of portable sampler options. If chilling of the samples is required, this is a passive cooling situation where we will either introduce ice into the sampler base to keep the samples in the bottles cool, or a popular methodology for that is to use gel packs. They have greater cooling capacity and they will last longer. We also have portable refrigeration options that can be AC or battery powered. And if we are using a battery, we do need to consider the amp hour rating of the battery to ensure that we have enough power in that battery to run the sampler throughout the duration that we desire to collect samples. The most common sampler option for this application will be the portable sampler. We also have bottle configuration options to consider. We have single or composite bottle configurations, and for COVID sampling, as Newsha indicated, generally we would be collecting composite samples. As this sampling routine becomes more and more utilized, there may be evolution where at some locations it becomes desirable to track the trend of the presence of COVID over a day. In those applications, there may be need somewhere down the road to look at multiple bottle sampling. The bottle material for COVID sampling that is preferred is polyethylene. However, that is not an absolute requirement. For the transport line, our standard vinyl suction line is okay, as is the standard weighted strainer. There are no issues of concern with interference with the materials for both those standard options. And there are no issues from the metal components within the transport line. As indicated, if the desire to collect samples will require flow-based or flow pacing, this would require an external flow meter or an integral flow module if utilizing the ISCO 6712. In short, something must be there to measure the flow to provide that information to the sampler. Other special features that may come into requirement: do we need start time triggers? In other words, there's a certain condition, certain event, or a delay down the road where we're setting the sampler up to date, but we do not desire to trigger into operation until sometime next week. Is there a requirement or a need where the pause and resume capability would come into play? In other words, if we're monitoring a specific location a specific facility that is only have active flow between the hours of, say, 6 and 5 o'clock p.m., and there's no flow after 5 till the next morning, we may want to configure the sampler or have a sampler that has the ability to be configured to only sample between those hours of the day and not sit there and spin its wheels when there's nothing to collect. If we're going to trigger the sampler into operation based upon certain criteria, certain thresholds, does the sampler have enabling capability that can be triggered into operation based upon those measurements? If we're doing sequential sampling, where we desire to collect a number of samples for each hour and then move to the next hour, that is a sampling technique called multiple bottle compositing. Do we need other parameters such as rainfall or pH, temperature, turbidity, conductivity, whatever, to be available and logged along with our sample collection? Is there a need for external control? In other words, we need some other device that is going to be dictating to the sampler when to collect the sample. Do we need telemetry? In other words, do we have needs from this application that we might want to have alarms, such as, I'm done sampling, so that someone can be informed that it's time to go out and collect the sample? If we're triggering the sampler into operation, we can set up an alarm that lets us know that the sampler is off and running. Do we have need for remote access to make program adjustments? Do we have the need to do data retrieval because we have collected information for other parameters such as pH and temperature? This would require the appropriate PC software to support these needs. The sampling locations for this purpose are not that different from other sampling needs. Samples can be collected in the sewer line that represent a segment or a particular area. Samples can be collected from a wastewater treatment plant influent. As Kaushal indicated, that there may be need to determine the effectiveness of the processes of that treatment, so we may want to sample at the effluent of the treatment plant. Some applications are surface water-based, rivers, lakes, streams. For COVID, at this point in time, those are not going to be too commonly utilized for sampling for this need. The approach criteria that are dependent upon contributing sources of flow can come into play. So for example, we may be sampling at a single location, i.e. the building level, where it's just that single facility that we're collecting samples to determine what's going on in that facility. Sewer lines, sampling there will give us a better idea about that particular collection basin, that particular area of town. And of course, the treatment plant is going to give us a more holistic picture of the entire location that that treatment plant serves. The population of the source has impact on this approach. And so if there are questions, it's usually a good idea to consult with your analytical lab for the best practices for any given application. If there are questions, always ask questions to make sure that those samples that are collected are going to be beneficial and useful. As with any sampling application, there are safety concerns, and this is no different. Normal practices should be practiced, such as wearing of gloves, masks, boots, to prevent one from having contact with the sample and one potentially causing some personal harm or impacting the quality of the sampler. And if entering a manhole, normal practices such as having a ventilator, air quality monitors, a winch to raise and lower one into the manhole, harnesses, and personal protection equipment.